So we're going to talk about Jurkat and a Microwell ELISpot system. And the goal of this research is just to optimize protocol and develop some reagents which allow researchers really to do this screening of multiple peptides against to identify and match T-cell receptor. And this one is very typical problem for any scientific research, because there are a lot of strategies to identify candidate peptides which can be used, for example, for development of anti-cancer vaccine or T-cell-based therapies. And it's usually start from the next generation sequencing, where you sequence DNA or RNA, generate a lot of bunch of the mutations or chromosomal abnormalities, or use mass spec, for example, and identify peptides which are associated with the cancer cells, and generate huge list of the peptides which need to be screened in order to see of their immunogenic activity, and at the same time to find matched T-cell receptors which recognize them. And in order just to do it, really, you really need to have some screening and validation peptide profiling pipeline. In order to facilitate these studies, we developed several technologies like shown here on this slide. And first, it's adaptive immune receptor profiling assay, which allow you to quantitatively characterize a level of T-cell receptor or B-cell receptors, if you want, in quantitative fashion, in bulk, in tissue or any cell samples. Similar assay was developed for single-cell analysis, which allow you to do analysis for sorted cells and allow you to get full-length chain pairing information and really expression of 30 markers for T cells. And last one, it's artificial cell system based on the Jurkat and K562 as antigen-presenting cells, where you can clone T cell receptors or peptide in order just to see how they interact to each other. Short introduction in bulk assay. It's multiplex PCR assay, where you design hundreds of the primers which matched all known T-cell receptor sequences. You can start from the RNA or DNA, you amplify CDR3 or full-length region, and you can get quantitative information. Importantly, really, it's incorporate so-called validator barcode. It's concept similar to UMI, which allow you to do error corrections and really normalization of any read number to the number of the starting molecule in any samples what you're using. And software, it's MIXR or platform, which is the most popular software used really in scientific community, actually free for academic researchers to analyze immune repertoire profiling. Single-cell analysis shown here, it's very similar concept. An idea to use it because when you just do experiment and identify small population of the cells which are activated, for example, based on expression of activation marker or peptide tetramer staining, number of the cells are not really very high. Usually it's maybe 100 to 200 cells. You don't need to run 10x genomics instrument to analyze them. You just sort them in 96 well plate. In each of the well of this plate, you have primers deposited on the bottom of this plate, which has barcode specific for each this well. You just, after you experiment sorting, you just put at -80 refrigerator this plate, and when you need it, you just incubate at 60 degrees for 1 hour. After that, mix all components of all these cells, wells together and run as one sample. It's very simple assay, but rather useful for chain pairing information and to get some information about phenotype of the T cells. And the last one, it's like I mentioned, K562 and Jurkat cell, reporter cell line. This one is classical system. A lot of people use it. It's already used for at least 20 years, introduced by, as far as I remember, Rosenberg from MCI. So, but this one is what classical system you use. You can clone anything what you want. It's completely not related to natural T cells. In K562, you can clone any HLA, and with the wake of the high throughput synthesis of genes, it's really so easy to clone any HLA. You can clone peptides as mini genes or fusion, like beta-2 microglobulin, HLA, and peptide, and you can create big libraries of peptides in K562 cells. In Jurkat, you can clone any T-cell receptor or, again, library of T-cell receptors, and if they interact to each other, they, like shown on this slide, they induce strong GFP activation, which can be analyzed by FACS. So in order just to use protocol, we did not invent anything. We just use one of the commonly used protocol for peptide, using peptides to stimulate normal healthy PBMC samples. And we just take MART1 peptide, which is rather well-known cancer-associated peptide, and grow cells for 2 weeks. At this moment, you can take samples at the beginning and samples at the end of stimulation, 2 weeks, and do quantitative RSA, what I described previously. And you can see here on the bottom of the slides, you can easily characterize, identify, based on the quantitative analysis, clonotypes which are expanded and after this peptide stimulation. In comparison with control, you have very good ratio between them. After 2 weeks, you can do re-stimulation with the peptide and using CD137 marker or staining with the peptide tetramer, you can identify full-length chain pairing information and really additional phenotype of the cells if you need it. Here's more complicated slide which show results what we generated in some sort of heat map. And first 3 columns here you can see it's in the beginning. It's control cells, DMSO control, and clonotypes, clones which are really significantly expanded in after MART1 peptide stimulation. You can see here expansion level of the clonotypes approximately several thousand-fold. And you can see this one's very clear. You can see very clear whether they're really specific, non-specific expanded clonotypes. Already from my point of view, if you don't want really to do very complicated experiment, it's already a good starting point and not to do any follow-up validation experiments. You can choose the guys which looks several thousand-fold already amplified in this after 2 weeks and just validate them to see what they recognize. But you can see here, next 3 columns, it's CD137, MART1, and interferon gamma staining of the cells after 2 weeks. And you can see here it's provide additional rather rich information, what clonotypes, but information is rather sparse. You can see here it's not very perfect correlation, but give you some sort of the confidence that these clonotypes, what are expanded, are real, and they are specific. Interesting, next column, which is prediction. You see these yellow guys. Yellow guys, we use algorithm from ImmuneWatch. It's machine learning algorithm which allow it to computationally predict, are these CDR3 sequences what we identify, are they MART specific or not? And you can see here, we have rather good correlation, but I would like to tell you the truth. This one, it's only work for the well-characterized probably peptides, which at least they have some reference data sets. If it's completely novel peptide, prediction efficiency will be significantly less efficient. So based on this result, we take 12 clonotypes which are upregulated in this experiment and cloned T-cell receptors in Jurkat reporter cell lines. We use HLA, which expressed HLA-A 0.201, which is very classical, most common HLA, and did by FACS, we analyzed expression of GFP. And you can see from 12, 10 are really very heavily upregulate GFP. The question is, okay, we run this classical assay. Can we make it better to generate more information and try to go to more high throughput? We just look at different technologies which are available right now in publications or on the market, and we find that this platform, which are produced by Celldome company, instrument so-called Clone Explorer, it's very cool and can be used for this so-called co-culture experiment where you incubate antigen-presenting cells and T cells together and look at the phenotype of these cells in order to see if they activate each other. And what core of this technology? Core of this technology, it's 96 well plate, where each well has 4,000 microwells, like shown here on the left of this slide. So you deposited your T cells, favorite T cells or PBMC, your antigen-presenting cells, and what good machine, this machine has completely all imaging, which can be at any time. And you use these plates in order to incubate in this instrument. This instrument allows do recording everything in time up to, and you can grow cells in this instrument without removing of the plate up to 2 weeks, because it's at the same time it's CO2 incubator, small incubator with temperature control. And it has also depth fluorescent imaging. And what, in order to detect secretion, for example, interferon gamma, or to do any immune assay, it's also very cool. They have deposited on the surface of the well antibodies against, for example, against interferon gamma. So if cells secrete interferon gamma, detection antibodies which present in the fluid, in the liquid above these cells, and cells start to be fluorescent. So you can immediately see if interferon gamma secreted. So you don't need to do any washing, anything, it's just online. And we did very similar experiment using this machine, where we just first get dendritic cells from monocytes. We just enrich the T cells for antigen using MART1 antigens and co-culture them in microwells in order to isolate hits. After that, we expand the clones in order to characterize them in more details using, for example, concentration dependence of the MART1 stimulation. And here's the result, how it looks. After first enrichment, we get about 100-fold enrichment of MART1 positive CD8 T cells. And we load antigen-presenting cells, dendritic cells, and T cells in this machine. And you can see here, we have very robust activation of the approximately 10 cells in each well. So approximately 100 different hits in this screen. In comparison with the control, we have very clear differentiation. This one's positive control. At the same time, we look at, because we look at kinetic, how they grow, and we sorted cells not only about expression of interferon gamma, but also based on how quickly they grow, in order to select the clones which grow well and for follow-up characterization. At the same time, we stay in with the CD4 and CD8 antibodies in order to see is it on the wells it's CD8 cells or CD4 cells, and selected them. This one on the right, if you look at it, shows how it looks real data on the machine. And you can see here's the beginning, it's one cell. When you load both of them 2 cells, you select the well where you have 1 antigen presenting cells, 1 T cell, and after that how they grow, clone. And you see here it's on the next interferon gamma signal, which you can see on the border of the well. And here's the CD8 staining at the end in order to select the wells with the CD8 cells. After it, you pick up the cells, which are, because this machine I didn't mention, they have picking some syringe which allow you to pick any colony and transfer it to 96 well plate for follow-up characterization. You see, when this clone grow, it's very clear, it's practically 100% MART1 tetramer positive. So here's the result, how it looks after that characterization of 10 clones. And you can see here, you can, using titration with different peptide concentration on the same machine, you can see you can get AC50 between 0.1 nanomolar to 5 nanomolar. So we show that it's really very rich characterization of affinities of these T cell receptors. The question is, this one is rather low throughput. Can we make it higher throughput? And here's the logic how to do it. You can definitely make synthetic TCR libraries, and we already hear even presentation at this conference where the people doing a lot of synthetic TCR libraries. They can be cloned in Jurkat reporter cell lines, or you can take a natural PBMC enriched for CD8. And in K562, you can again clone peptide library and download them in this machine and screen for the interaction between one library against another library. And here's the example how it can work. We just built library in 120 cancer-associated peptides, clone them in lentiviral vectors in K562 HLA background cells, and screen them against CMV T cell receptor clone in Jurkat reporter cell line. And you can see after several days of growth, we select the clones which are positive for secretion of interleukin-2 and GFP positive, and to see that in majority of the cases the CMV peptide. So it shows the power of this machine and this strategy not to screen one library against another library. The probably maximum complexity for one plate, it's 100,000 combinations. So it can be, for example, 1 against 10,000 peptides, or 100 peptides against 100 T cell receptors. It's just up to you really what you can do in this type of the experiments. And last slide, it's a summary what we discussed today. We discussed today development of several technologies which allow you to facilitate this type of the screening and some ideas about strategies how these protocols can be used for large-scale screening of peptide library against T-cell receptor libraries. Thank you very much for your attention, and looking forward to answer your questions.